Good afternoon and welcome to today's Friends of Cancer Research virtual meeting, Shaping the Future of Emerging Immunotherapies and Cell Therapies. If you don't know who Ned Sharpless is, then you probably joined the wrong webinar. Ned is a man that needs no introduction at all. As director of the National Cancer Institute, Ned continues to lead research initiatives that are the forefront of innovation and cancer care. The NCI has continued to recognize the transformative potential of the breakthrough therapies we'll be discussing today. Their work is extremely important to patients and researchers. Thank you, Ned.
Hello and greetings from the National Cancer Institute. It's my great pleasure to speak to you today. Friends of Cancer Research has been doing such great work for so long that I truly think of you as partners in this common struggle of ours, this fight against cancer. And the subject of today's meeting, immunotherapies and cellular therapies, is really an area of research that now sits in a kind of sweet spot, if you will, a real nexus of great potential validated by real progress. And it's a very promising time in this field. We're seeing new approvals of FDA approvals of new immunotherapies, and a really robust variety of cellular therapies making it in the clinic, and how to combine these agents is an exciting time. And as you know, the National Cancer Institute has long been at the forefront of the cancer immunology field, whether that's by the work of people like Steve Rosenberg in the intramural program, or the exciting science by many of our extramural fundees, people like Jim Allison and Carl June, who really been so important to these fields. So these fields have advanced rapidly, producing several new methods that increase the strength of the immune response against tumors. Progress has been particularly noteworthy for hard-to-treat cancers like melanoma and lung cancer. And of course, these advances in cancer immunotherapy, they really are the results of decades of long, difficult investment in basic research on the immune system, much of which has been supported by the NCI. Improving and expanding cancer immunotherapies is a key research priority for the Cancer Moonshot. As part of the Moonshot, NCI has established two networks to accelerate the translation of immunotherapy research discoveries, the rapid translation of clinical applications for both adult and childhood cancers. Among the Moonshot supported groups working on immune-based approaches to treatment in adult patients is the Immuno-Oncology Translational Network, or the IOTN. Their efforts have led to insights into the response of ovarian cancer to immunotherapy and into the use of immunotherapy in certain types of head and neck cancers. Another effort is developing an immunotherapy research network focused on childhood cancers. And so the Pediatric Immunotherapy Discovery and Development Network, or the PIDDN, has been studying major barriers to the development of immune-based treatments for children and adolescents. So a recent study from that group, supported by this network, suggested that some children with B-cell acute lymphoblastic leukemia that recurs or resists treatment these patients may benefit from certain immunotherapy type approaches using drugs to target the immune system. Early last year, NCI turned an existing virus production suite at the Frederick National Lab into a CAR-T cellular manufacturing facility and began supplying manufactured cells to multiple clinical locations for early phase trials in a multi-center way. Also coming online this year at NCI's Bethesda campus will be a brand new facility for production of tumor-infiltrating lymphocytes, or TILs, a technology really invented by Steve Rosenberg. Despite all of this progress, in many ways, this approach is really still in its infancy, with many challenges still to come, such as how to optimize the immune responses to eradicate cancer while avoiding runaway immune responses that would damage normal tissues, and how to identify patients and types of cancer that are most likely to respond to immunotherapy drugs and overcoming the resistance to these agents. How to increase the effectiveness of immunotherapy by combining it with other types of cancer treatments, such as targeted therapies or chemotherapy or radiation therapy. How to improve the safety of immunotherapy by understanding why some patients develop such toxic side effects and how to predict these effects and mitigate them. And we're really aware of the regulatory challenges inherent in some areas of immuno-oncology, such as highly personalized vaccines. And we're really committed to working with our federal partners, for example, at the FDA, to address these regulatory challenges. In fact, the word partners will always be a feature of any progress we make, not just with the FDA, but with all of those who occupy the broad spectrum of research needs, from the needs of funding to those of research talent, to the needs of public education and outreach and advocacy. The importance of partnership is exactly why organizations organizations like Friends are so important. Convening events like these to share our challenges, our advances, and our potential solutions and ideas on how we can move the field forward. So again, thank you for your great work. I know this event will prove very critical in our common goal of progress against cancer.
Good afternoon. I'm Jeff Allen, President and CEO of Friends of Cancer Research. Thank you all for joining us today for this exciting event. And thank you, Dr. Sharpless, for sharing your perspectives on the future of cell and gene therapies as we kick off today's meeting. Over the last several months, we've had a group of leading experts working together to develop the white paper that you received in advance of today's event. The collaborative nature of this work speaks to its importance toward optimizing the future of immunotherapies. We hope it will serve as a blueprint of sorts for ways that we can work together as a research community to identify and mitigate emerging toxicities earlier as novel therapies are developed. I'd like to thank the working group for their time and expertise and turn things over to several of the group members, starting with Bruce McCall from Genentech, who will be leading our first panel. Bruce?
Thank you, Jeff. I'm Bruce McCall. I lead the clinical safety group for the hematology portfolio at Genentech Roche. And to set the stage for today's panel discussion, I'd like to make a few opening comments. If you could bring up the content, please. So today we're really going to be focusing on harmonizing the definition of cytokine release syndrome in clinical trials. Next slide. The issues around this is, as the doctor previously mentioned, there's been transformational advances in cancer immunotherapies, and that's brought awareness of novel toxicities such as cytokine release syndrome. We first saw this, for instance, with immune-related adverse events with checkpoint inhibitors, and now we're seeing other toxicities with cellular products and T-cell engagers. We know that CRS is a critical feature of the benefit-risk assessment of these promising therapies. The approved products to date all have black box warnings around cytokine release syndrome. And the transformational nature of these therapies has led to an explosion of rapid development that was described across academia, across investigators, across biotech pharma. But with that explosion of development, therein lies the challenges in harmonizing this landscape. For instance, among the 4 approved therapies, 3 cellular, 1 T-cell engager, there are 3 different CRS scales in use, and thereby really 3 definitions of cytokine release syndrome. Next slide. This dilemma also includes the difficulty in distinguishing cytokine release syndrome from infusion-related reaction. This obstacle was not first appreciated with the development of CAR-Ts, in that CRS is delayed from the time of infusion until T-cell expansion, but essentially was unmasked with the development of T-cell engagers in the clinic, where the time of onset overlaps typically with infusion-related reactions in CRS. IRRs are typically described as occurring during or within 24 hours of infusion. And this issue extends beyond simple nomenclature because the discrimination between these 2 diagnoses impacts the prognosis, monitoring, and management of the respective issues. We predict that this is going to be an ongoing challenge with novel IO agents and targets of the future. And the white paper provides a developmental model by which early clinical programs can approach this diagnosis and designation issue. Next slide, please. So what we're really here today is to have a call of action for the collective us to have a harmonized definitions and reporting of cytokine release syndrome. We'd like to emphasize the importance and value of critical elements of the case report form of clinical trials so that we can map data towards future scales if those CRS scales or definitions are needed, and to equally important, to adequately characterize the patient path from time of presentation, diagnosis, treatment, and resolution of CRS. And all of this will require the collaboration and partnership across pharma, across academia and the health authorities. Next slide, please. So as Jeff mentioned, I'd like to acknowledge the efforts of the working group. This represented a broad group of people, key members of the FDA, the NCI, academia, as well as a broad representation from biotech pharma. And now I'd like to introduce our panel. Meredith Chuck from the Center for Drug Evaluation and Research, FDA. PK Morrow from Amgen. And Marcelo Pasquini from the Medical College of Wisconsin.
Thank you, Bruce. Happy to be here.
Thank you all for joining. My first question I'd like to direct to Meredith and PK, and that's, how would you briefly describe the advancements made with IO and cell therapies and their expanding use in treating cancers, the breadth of applications, the novelty of the platforms, and most importantly, the impact to patients? First, you, Meredith.
Absolutely. So thanks, Bruce, and happy to be here. So, if you look at the broad category of immuno-oncology drugs, it really is hard to overstate the impact that the development in the last couple of years has had in changing, clinical treatment of patients with cancer. If you start with the immune checkpoint inhibitors, we have FDA-approved indications for everything from melanoma to lung cancer to lymphoma, GI, GU, renal cancer, and several tumor-agnostic indications as well. More the focus of this meeting are the emerging immuno-oncology agents. So looking at the CAR T cells, there are 4 approved agents in lymphomas and pediatric ALL, and a number of ongoing drugs undergoing evaluation, and also lymphomas and a number of heme malignancies. If you look at the T cell engagers, specifically the bispecifics, there's an approval in ALL, but really a very robust development plan in a number of hematologic and solid tumor malignancies. So, this really has been a very robust development program. It's probably only going to get more so in the coming years. So getting things right, like identification of toxicities, consistent data management, so that we're really able to inform patients and providers is key.
Thank you. And PK?
Yeah, Meredith, and that was great. So just building on that, when we really look at the significant evolution in our understanding and development of these immuno-oncology therapies, we acknowledge the evolution of the response criteria for IOs, and we really understand how far we've come from really better understanding, for example, what pseudoprogression is, to integration of our understanding of response criteria into our modified iRECIST. It's really clear we've come very far in developing IO therapies. What's now just as important, and is at the other side of the coin, is ensuring that we have a robust and a shared understanding of the toxicity profile of these agents, which is why we're so excited to join this working group and contribute. Ultimately, if we don't contribute to understanding CRS across this industry and across patients and caregivers and understand how to grade it, we won't be able to appropriately advance drug development and won't be able to advance cancer care.
Thank you both. My next question is, because this is such an emerging field, there's understandably a huge focus on the safety profile of these novel agents. What can we do, FDA sponsors, academia, what can we undertake to maximize the understanding of these new therapies, particularly not only in early development, but in the post-marketing section? And Marcelo and Meredith, if you could please, in that order, take on this question.
Yeah, sure. Thank you very much, Bruce. So in the post-market setting, I think the major effort is standardization. And standardize because now we recognize the features of these of this syndrome. For example, these CAR T cell or IO specific side effects. And but the IO standardization should not be rigid. We should allow ourselves to have some flexibility to understand certain phenotypes that are peculiar to certain CAR T cells. Now we recognize CRS as an identity. As a toxicity identity that includes several features. But as new agents come up around the pipeline for us to understand them, then there'll be additional elements that may incorporate into the CRS. So looking at standardization is really a big effort for us to understand. Now, in the post-market setting, where we see a much larger number of patients, we can start seeing some granularity on phenotypes that may inform the preclinical setting. So then we can build this in a cycle of development. So that would be a very important element for what we learn with the post-marketing setting, with very rare events, inform the pre-market setting, so we can really refine the CAR T cell or other IO-specific toxicities.
Yes, the post-translational effort going back to the preclinical space is something of vital importance, and certainly Genentech, and I'm sure others think a lot about. Meredith?
Great, thanks, Bruce and Marcel. I completely agree. I'm going to step back and go to the pre-market setting and just talk about standardization from a regulatory perspective, how critical that is to do in those early phase protocols. So taking that preclinical information, taking information from similar drugs in class, trying to anticipate what the toxicities might be, and then designing protocols, patient management, mitigation strategies, and collecting the data that really allows those interventions to be data-driven. This is especially critical for a lot of these expedited development programs that we're seeing, where those early phase trials often become the registration trials. So what ends up in our label often is what was done in those early trials. So trying to standardize those common elements among the drugs in class will really allow us to get some evidence-based driven modification strategies, dose modification strategies to put in the label so that patients and providers are really able to adequately assess the benefits and risks. And one comment about your post-market additional refinement, that's key, but I think the key is also you want to define your toxicities in the pre-market and refine them in the post-market. We don't want to get into the sense where we're defining toxicities in the post-market because the data is never quite as robust.
Excellent, thank you. PK, and then Meredith, maybe you can tackle this next question. Where do you see the advantageous elements? What are the advantages of really this cross-pharma, cross-academia, cross-regulator partnership in moving standardization forward so that ultimately we can better characterize these things across pipelines and better help patients?
Thanks. So let me start, Meredith, and I know you have a lot to contribute here. So, despite the fact that we've talked about how much we've done in immuno-oncology, I still believe that our IO field is still in its adolescence. As we continue to build our understanding of cytokine release syndrome and the other complications of IO therapies. And so currently, as many of us review data and attend conferences and discuss data, it can be increasingly difficult to adequately compare safety data across programs, making sure that we're comparing apples to apples, given the differences in the CRS criteria and the data capture elements. Thus, it's really, really critical that we arrive at a harmonized approach towards grading CRS, as it highly impacts the potential for appropriate CRS mitigation and prevention ultimately, and it can also affect dosing. And when we talk about that, we need to ensure that this approach is a modular one, building in the components of not just the clinical aspects that are particularly clinically relevant and require mitigation, prevention, and treatment, but also understanding and appropriately harmonizing across not just industry, but with academic investigators and particularly with our regulatory agencies. I'll turn it to Meredith.
I couldn't agree more. I mean, I think the consistency in data collection and definitions is really critical, not only for regulatory purposes. Obviously, that streamlines review. It allows a little bit of cross-trial safety comparison, which is a little less fraught than cross-trial efficacy comparisons. But it also helps to inform, again, product labels in class that will likely lead to similar risk mitigation strategies. It also allows for consistent conversations with patients about toxicities if that data was collected consistently. It also allows for research endeavors to be able to look at similar data elements across trials. And then eventually, if once that data becomes in peer-reviewed literature as well, it also allows for the public to be able to look at those events similarly across trials of products in the same class.
And Meredith, to follow up on perhaps a little bit of a provocative question, in the pre-marketing space, where would you judge the harmonization or disparity as of today?
I'd say we're getting better, but there's certainly work to do. I think common data sets, presenting data standards, potentially common electronic case report forms. I mean, those are some of the agreeing on those data elements, agreeing on definitions for them, agreeing on how to present that data. I think obviously would require a lot of pre-competitive collaboration, but I think that's really what's needed in this space with the vast majority of drugs and only going to increase.
Yes, I think we're all actually trying to make the case where everybody's a winner with that collaboration, the patients most of all. Marcelo, let me direct this next question to you. Of the many hats you wear, one of them is being the registry director for a large cellular therapy registry in the United States. From that aspect, perhaps, what are the downstream benefits of doing harmonization now for patients, for regulators, for developers?
Yeah, thank you, Bruce. The benefit is that when you define a critical elements, or really it's a common denominator that can really serve multiple stakeholders. So the approach is to define really the main criteria that, or the elements that define the particular syndrome. So for example, for CRS, you collect the elements so then you can define not only the onset, but also the severity of this. And I think we did spend a lot of time in the white paper to really expand this in a way that as the field evolves, there's more information that comes through it, which is not only defined on specific products that are available now, but future projects that have really focus or target other cells, cells or tumor histologies. So really the benefit of that is to really create a stepping stone for us to do analysis in the future. And for multiple stakeholders, having this sort of information, it really, you don't have to reinvent the wheel every time there's a new product. So I think that is really the most important aspect of it. And I think, again, what we really learn in the post-market setting really should inform the pre-market setting as well. I think I do respect Meredith's point that you shouldn't really reinvent the wheel in the post-market setting. But I think elements like one of the examples that we really are coming to understand now is really severe forms of CRS, like for example, the HLH or macrophage activation syndrome, that really we need a lot of patients to understand really what phenotype comes through. So really this is, once you have the baseline of critical elements, really this is really helps us to make these products safer for patients.
Yeah, that does speak, for particularly rare events, that does speak to the advantages of collaboration, even in the pre-approval space. So thank you for that. And now opening this up to all of you, could you spend some time commenting on a few things? What in the white paper stands out for you the most as really essential elements of that? And maybe you could further comment on the proposals for data collection harmonization. Why will that really make a difference in the future? And as PK pointed out, we're here now in its infancy in some ways. How can we seize the moment now to really impact things downstream? And whoever wants to start, please go ahead.
Well, maybe I could start. I think that, this panel has been spot on in terms of its guidance. And, one area that we need to kind of focus on or maybe have a thought about is that while we've always need established monotherapy efficacy, a lot of our treatment paradigms are actually involve combination therapy. And in the future, that combination therapy is likely going to include 2 or more IO therapies in combination. And so it's really going to be even more vital to collect our data in a way that's granular enough to allow us to pivot and adapt to our evolving understanding of the toxicities associated with these IO therapies as we try to tease them out. And as we were developing the white paper, we tried to be as specific and detailed as possible to support this harmonized approach. But really, I believe the white paper is just the beginning. We need to continue to iterate and build on this learning to enable us to also, as Meredith was pointing out, build the eCRFs, build the data elements, and completely be harmonized there to enable us as we continue to advance drug development, allow us to better understand the safety profile of these therapies. And that goes beyond just the ones that we know today.
Thank you, Meredith.
I'll take that next. So thanks, BK. I couldn't agree more. I think one of the elements of the white paper that stood out most to me was the call to collect data to the granular level enough that you could both uncover the individual components of a syndrome such as CRS, but then be able to collapse them down at the end once you have more robust data to essentially call it that syndrome. So I think it's really critical to be able to capture the fever, the hypoxia, the hypotension, and then be able to link that back to the CRS event itself. I think also critical is really being able to link any interventions during the clinical trials for the patients and link the outcomes for those interventions. Because again, as we're developing risk mitigation strategies, we want those to be data driven. So we really need to be able to build a story from the data that we're collecting in the clinical trials. And I think if we do that in a robust way, really the ultimate goal, I think that everyone is striving for to maximize patient safety is to be able to do predictive models. So we want to be able to know which patients are most likely to not only benefit from therapy, but have the toxicity so that we can potentially tailor some of those mitigation strategies for those particular patients. But we're never going to get there unless we do very detailed and granular data collection early on in the development program.
Yeah, and,
yes, a personalized approach would be a game changer. Marcelo.
Yeah, just the final moments here. I agree, and I think this this exercise or this coming together as a collaboration was very helpful for us to start thinking about other toxicities, thinking about safety. I think looking at all the members of this group really led us to view this problem in different perspective. And I think building on making these products safer is really a hallmark here. And I thought this was a very successful exercise. I'd like to thank the Friends of Cancer Research to put this together.
Thank you, all of you. As mentioned, really, this is hopefully only the beginning. And I think all of us look forward to further developing these ideas and working towards that collaborative spirit. With that, I turn it back over to Jeff.
Thank you, Bruce, and thanks to our panel and to all the working group members. I hope that everyone has the opportunity to go through the white paper in depth as it provides an important considerations for the early development of novel therapies, as you've heard from our panel. Next, to look to the future of cell and gene therapies, we'll move to panel 2, and I'll turn it over to Anna Edney from Bloomberg News. Anna?
Thank you. I'm really excited about this great panel. We have Peter Marks, the Director of the Center for Biologics Evaluation and Research at the Food and Drug Administration. We have Renu Vaish, the Vice President of Global Regulatory Affairs at Kite Pharma. Jennifer Malin, the Chief Medical Officer, Oncology and Genetics at United Health Group. And Mark McClellan, the founding director of the Duke Margolis Center for Health Policy, and also a former head of the FDA and the Centers for Medicare and Medicaid Services. I wanted to, thank the last panel. I think they had some really great detail on a lot of this, and I wanted to take a step back, maybe for someone like me who isn't at a drug maker, who might not know as much about some of this, and just ask Peter, if you could give us an idea of this emerging field, where it was 5 years ago, and how it's changed in those 5 years, and how the FDA is changing to try to adapt to dealing with what I assume is an increase in applications.
Yeah, so thanks very much. Thanks for tuning in today. So CAR T cell therapies have really taken off over the past few years. Let me just start with just the numbers, which is that, in 2015, we had about 20 INDs submitted. And last year we had 58 INDs submitted in this area. And the past 3 years, it's been increasing numbers of CAR T cells each year. And the shift now is towards seeing more of these in later stages of development, phase 2 or 3 studies. Importantly, what's happened over the time between 2015 and now has been a real maturation of the manufacturing process of this. In 2015, we were, before we had approved products, we were still working out reproducible manufacturing, scale up, being able to do autologous CAR T cells on a regular basis. And we were not even that far along towards the allogeneic CAR T cells, which are now well under development under IND. So a lot has happened, a lot of innovation, and each year brings new introduction of technologies into the CAR T cell world. The past year or two has brought the allogeneic CAR T cells into play, particularly for solid tumors, where people are now starting to make multiple changes using genome editing to help make products that could potentially address solid tumors, in addition to the hematologic malignancies that have been, some of which have been very well addressed by CAR T cells. So very rapidly evolving field, and I think a lot of room to grow in the future.
Rainu, I wonder, we heard a lot about standardization from the last panel, and I wonder if you would agree that seems to be kind of one of the biggest challenges in development, and if there are other things that you might point out that that could be helpful.
Sure. Good afternoon, everyone. Glad to be here. Yeah, no, that's actually a very important topic that we are exploring, is the standardization in terms of the CMC specifically. So as we think about our programs, these are global programs, we are working to make sure that we could address some of the potencies and the specifications across the globe as much as possible. So if there is a standardization that we can use on that aspect, that is certainly helpful. I think also, there was a great conversation in the morning aspect of the data collection and the standardization. So I completely agree about where it is that we want to go. I think we're getting there. I think part of the continuous build of these novel therapies and access is allowing for that. But the work is still there to continue.
And, Mark, some of this is I think something that, people like that have been in your position have thought a lot about. And I wonder, what are the emerging kind of best practices to handle some of this?
Well, I think what we've been talking about today are exemplifying the best practices, and I really want to congratulate Friends for moving this approach to standardization around CRS, and hopefully a path to standardization more generally forward. As you just heard from Rainu, there are challenges in presenting data and in understanding data to help get through the regulatory process, not just in the US, but around the world. And I would just emphasize too, I know you're going to go to Jen Malin in a minute, that it's important for the developers to consider payment and post-market developments as well. And that's where these kinds of standards can be really helpful for CAR Ts, for other therapies in this immuno-oncology space. They are complex. They involve, as Peter knows well, challenging cellular processes to regulate and understand. And the good news with that is that we are likely to keep getting better, but we're only going to keep getting better at understanding potency, understanding patient response, and understanding toxicities like CRS and how to avoid them if we are collecting a lot more data in actual use. And that's where I think these kinds of standards can make, there already are a number of requirements for post-market monitoring and the like, but we want to make those as effective as possible with data that can be interchanged, data that can be analyzed, data that can really help us understand better which particular projects or products are best, how they can be targeted, and how important complications like CRS can be avoided and managed when they do occur. So lots of opportunities for moving the field forward by building on the kind of approach that that Friends is taking today.
And as you mentioned, I do want to ask Jennifer about payers and what could be useful for payers to ensure access to cell therapies, and it sounds like standardization obviously is a big one. I think.
Sorry, I was on mute there. I think that the challenge for all of us with innovative therapies is we don't know what we don't know what's not measured, right? So as new therapies are developed, and if they have new side effects, new toxicities, if there isn't kind of at the front end, you're not even necessarily capturing them. And I think that's as everyone's been saying, what's so terrific about the work that's being done here is standardization, but we're now 5 years into these therapies being on the market. So, from a payer perspective, we're not involved in pre-market, only post-market, right? And then we're trying to figure out how do we introduce this to our members in a way that ensures that they have that novel toxicities are being addressed. So typically we look to provide synergy with the efforts of the developers and physicians who are trying to treat their patients by limiting initial access to centers of excellence. This allows us to provide requirements that they participate in data collection, that they meet the standards, and that they'll be closely involved with developing treatments to mitigate the toxicities. And I think that's one of the early benefits is that we all want the registrational trials to include accurate measurement of toxicities, but with novel therapies, we often find that as they get used, that the toxicities may decrease as people get more comfortable with managing them. Even with something as basic, say, as irinotecan, which is an old chemotherapy drug, it carries a black box warning because in the early trials it had horrible diarrhea, but after it'd been on the market for a year, providers figured out how to manage it, and it's really not that much an issue anymore. And so, I think this is an evolving and iterative process, and I think we're always looking in the rearview mirror to figure out how to anticipate the next innovation and what can we do better next time.
Right. I was kind of curious as Jennifer was talking, actually, how do the drug makers kind of feel about limiting these to centers of excellence? Does that make your job post-market tougher, or is that a better option?
Yeah, no, it's a great question and a fair one. So, as part of cell therapy and gene therapy, we have requirements for long-term follow-up. And so I think the 15-year long-term follow-up, the potential of post-marketing registries, how can we harmonize these into something that's going to be successful without having to also carry incredible infrastructure aspects of setting these up? So this is something that's top of our minds. It's actually something we talk about regularly. And if there is an option to consider how we could do these more effectively and efficiently in terms of the post-marketing safety, as Jennifer said, it's been 5 years, there is some experience. Is there an opportunity to potentially decrease that or make it more efficient? Because that does have an impact on the sponsor side.
Peter, I think obviously everyone would probably appreciate sort of where the FDA is going at, with a lot of this. Is there any effort? I know we talked about the standardization of kind of the terms and things, but is there something else that also FDA might be looking at in terms of review or manufacture or anything along those lines that could facilitate development?
Well, I think we're trying to figure out ways that we can have, there are several different aspects of this. Obviously, we'd like to be able to have sponsors be able to make changes that are beneficial to these products without having to fully recharacterize when they're below a certain threshold. We're going to try to get better at understanding where that is, because these are therapies that are likely to evolve with time. So I think that's one place where we're looking to do work. I think then also how do we best understand the best ways to clinically develop these will be another. But I think the place where I see the most interest right now is really around characterizing manufacturing changes. How do we allow these products to evolve in a way that we're comfortable at FDA that that we have enough information to make sure that people will be safe, that they're being treated with quality products, and yet we don't hold back the pace of development so that new technologies don't get incorporated. I mean, the problem here is you don't want to have to wait 5 years to have to incorporate something in a field where every year something new is changing, because then you're basically always have essentially stale products out there in the marketed zone. So there does have to be a way to improve the cycle time. I don't have the perfect answer to it, but it's something we'll have to continue to work towards.
And I think that this is part of sort of the risk benefit analysis that FDA is often doing. And, there is sort of the personalized aspect of this. And, Mark, I wonder if you could talk about that a little bit as far as some of the specifics for cell therapies that are weighed, that are maybe different from other drugs that the FDA is considering.
Well, I think it does get to the issues Peter was just discussing. Many of these therapies are complex. It's not as simple as a small molecule drug, or even in many cases, a larger molecule biologic. And that does mean that there are likely to be opportunities, as Peter said, in the production process to improve or further optimize products that have made it to the market. So I think it's really important to have a clear understanding and on how manufacturing changes, production changes can occur. But I would expect that there's some hope that and some benefits that FDA can take, patients can take from the fact that, coupled with a careful and thoughtful but timely process for encouraging modifications and improvements, is a very good, reliable process for noting what the effects of those modifications are in practice. And that gets right back to having very reliable, standardized, and well understood, well characterized data in these post-market registries, like the one that Marcella was describing in the last session. The more that people can have confidence that those registries are complete in terms of detecting safety issues, the more that people can have confidence that they are reliable in terms of characterizing those issues, I think the easier it is to have a whole regulatory framework that supports further innovation in these complex products.
Jennifer, can you give us an idea of what payers are looking for in these type of registries? I know you mentioned data collection. I just wonder how you're using that information.
I think, it gets used, I think, in two different ways. I mean, the first is, as these new therapies come to market, there's a need to figure out how to pay for them. And the cost to the payer includes not only the product itself, but all the services that go into delivering the therapy. And to the extent that there's side effects that require hospitalizations or ICU care, that can have a big impact on the cost. And so figuring that out, is part of bringing, of providing access to people, because if the cost of delivering the therapy isn't covered, hospitals aren't going to be, that excited to provide access. So that's a really important piece early on. I think over time, as there's, we heard there's 58 INDs, which is, this past year, that's really exciting in terms of new innovation and new, potentially new patient populations being treated. But it also likely means that there's different products on the market, and that over time we would want to use this data to both identify which is the most effective and safest product, but then also potentially use that information to try to decrease the very high costs by having some competition between the manufacturers and negotiating lower rates. And you can't do that very effectively if you don't have good data that tells you that the products are equivalent. And so I think those are the ways that, over time we would use this to, to maybe bring to the therapeutic space more the kind of competition that we've seen in some other technologies, like, our cellular phones, our personal phones getting not only better, but lower costs, which we haven't really seen happen in the pharmaceutical sector.
Can I just add one more comment here? I want to agree with Jen's point about how better post-market data is going to encourage and support further innovation that's going to benefit patients for all the reasons that she described for the products. But I think that also is true for complex products like this, where administration needs to be managed carefully and side effects are going to occur, is to encourage, frankly, competition and excellence in the providers, the hospitals, and the specialized centers that are delivering these services. The closer we look, the more we see variation. The more complex the process is, the more variation there generally turns out to be in terms of the ability and experience of hospitals or centers to manage both targeting patients effectively, delivering the treatments effectively, and especially managing some of these complications that will occur. And probably the model for many of these therapies early on and maybe even later on is not that they should be done widespread at every center, but if we want to get the most value and the best outcome for patients, is really have some expectations about centers that are delivering these services to be able to track outcomes and complications in their patients using these more sophisticated and more precise and well understood data systems. That's what you'd like to have payments to support. And I know Jen and others work on centers of excellence models that are like this, and I see maybe some future developments in the direction of sort of whole packages of service delivery for these patients that includes the hospital administration of a CAR T, plus careful screening and prep beforehand and follow-up that's really part of a overall more coordinated care model that probably is going to be paid for differently in the future than just CAR T payment for a hospital admission. These kinds of data systems make it easier for those approaches that are intended to increase value for patients, getting them to the best possible place to get their CAR T therapy or other immuno-oncology therapy. And it really will encourage, hopefully, a race to the top among providers of these services, not just more of them trying to do more of the latest IO therapies, but more of them really trying to specialize in doing the most important and most impactful therapies well with fewer and fewer complications.
I think that's a great lead in to kind of, and you hit on a lot of, what the future of cell therapies could look like. And, one of the things I wanted to ask Peter is when you talked about these, not wanting to hold things up for 5 years while you try to figure out, the next step. When might people who are interested in these expect kind of more clarity or guidance from the FDA on something, on how to handle updates or things like that. And when you're moving so fast, like these therapies are.
So I think you'll probably see us say a little bit more about this in the coming months, or maybe even sooner. And we'll continue to work on this. I think this is an area where even guidance that we issue will have to be continually updated as we refine it and as we learn more. So it's an area where we'll continue to work, continue to listen. So I would say that even if somebody sees something in our guidance that they are not fully happy with, just know that it will likely continue to evolve.
I'd love to know too, kind of what that's sort of the future of where FDA is moving on the regulatory front. Where do you see kind of the future of cell therapies in general going? What breakthroughs do you see coming?
Yeah, so I think the big issue for cell therapies, whether they be autologous or allogeneic cell therapies, or that for that matter, other cell therapies outside of lymphocytes, is probably going to be understanding scientifically how to characterize them better. This is kind of a dry, geeky thing to have to talk about, but there's the manufacturing process that is challenging for these products, and it's made all the more challenging because we don't have good methods of characterizing what's happening. Manufacturing is a little easier when you can analyze what's coming out the other end. That's why when you make small molecules, it's not, if the reaction isn't perfect, you can look at, you have analytics on the back end that are known, trusted, and you can figure out what's going on. The problem in cell therapy is if something doesn't go exactly right on the manufacturing, it's much more challenging. I mean, you can figure it out, but sometimes it's much more challenging to figure out. And sometimes you can figure out what's wrong, something's wrong, but you can't figure out necessarily how to fix it. So having the analytics and having those developed may be a very important piece here in allowing us to make progress. Certainly, it's a little easier perhaps on the allogeneic cells because you can work with them and you have plenty of more time to work with them compared to the autologous cells where you've got one shot and you have to have a process that leads to a good product. And if you had better analytics that you could use in real time, that might actually help to have better products for overall, both for the allogeneic and the autologous products.
And Rainu, I sort of wanted to give you the last word here on what you see as a breakthrough coming and what you see as the future of cell therapies.
Yeah, no, thank you. I just want to comment a little bit on what Peter said. I think that is actually the number one thing that we are also going through. So the innovation, where we see this, we are exploring very novel INDs where, one of the advantages we have taken, of course, is the opportunity to seek FDA feedback. But, I really resonate with what Peter said, because that's exactly what's happening is you do the work and then you realize that the output is not exactly what you expected. So there is a learning curve as we continue to develop these very, very novel areas, both in auto as well as allo. And then now we're talking about potential combinations with auto and allo. So then you've got another sort of area of development. I think in terms of breakthrough, I think the fact that if we could try and understand how we could lower perhaps the long-term follow-up, the clinical studies, I think, are very clear. I think where the development is going to come through is on the CMC side. And if that is something that we could develop universally.
Thank you, everybody.
Thank you, Anna, and thanks to all of our speakers. Wow, you've provided a great overview of the complexity and the promise for the future. So thank you for your insights and for your continued leadership. This concludes our virtual meeting, Shaping the Future of Emerging Immunotherapies and Cell Therapies. I'd like to take another moment to thank all of our participants, working group members, and panelists for their important work on these topics. I'd also like to thank the Friends staff for their hard work to put together these events and lead these timely projects. As you saw at the top of the event, we're celebrating our 25th year and have a great lineup of events and future initiatives as we celebrate our work and plan for the next 25 years of our organization. Please stay tuned for more details, and please reach out to us if you want to get more involved with our work or our 25th anniversary programming. Until next time, thank you and take care.